plankton tow. What we used for this was a net. We used a net to catch the plankton tow, and that was connected to the bottle, which held all the plankton tow in it. And what we did was we'd set a distance and we'd walk back and forth with the net, and we would have people recording other data. But what we did with the net was, after walking back and forth, we'd have someone lift up the net, and we couldn't just set it down in the water quickly because you don't want air in there. But after they let it up, you're gonna have to rinse it off with the water that we tested all the other stuff in, the temperature, the pH level. You're gonna take that and we turn the net, and they just rinse it off so all the plankton inside of the net could go down into the bottle, and then we would just put the lid back on the bottle. For the wind speed, we had to go in a 360 direction to test the highest amount of wind speed. We tested the amount of salt in the H2O. So what we took a tool and we had to use it to look up the sun, but first we put distilled water on it for a base, and then we rinsed it off with the salt water, and then we put some drops of salt water on it, and we looked up at the sun, and it helped us tell the percent of the salt salinity in the water.
They were collecting this data, the researchers. So we worked with two different women who lived there pretty much full time and collected this data on a daily basis. At least that's what it seemed like. So there was Lynn. She coordinated all the teams that would come to the island and helped them collect data and worked with them basically the entire day. She was with us all the time. There's also another woman named Linda. She did the same type of job, so they collected data and worked with all the student groups that came through their island. So the first day they taught us how to do all these things. So not only how to do the plankton tow, but then how to take that plankton back to the lab. So what we do is each day we'd collect the plankton, we'd take it upstairs. So they have a big lab with lots of microscopes. We'd all put on lab coats, which made us look really very professional. And we would take a pipette and we'd collect a little bit of that water with the plankton in it and put it on it was about this size with lines through it. So when you put it under the microscope, it was labeled 1 through I think maybe 25 or something like that. Yeah. And so you could see which plankton were in which spot just to be able to find it easier. So what we did is the first day we just looked for any plankton. So plankton is anything that is freely moving in the ocean. So it can even be things that are going to grow into much larger things like crabs. Like there was tiny little plankton, because they're called plankton because they just get moved by the waves, but little tiny larvae that was going to grow into something much bigger. So the first day we just looked at everything that was in there. So there's two things. There's zooplankton and phytoplankton. Zooplankton is the most fun to look at because it's little tiny animals. Again, things like crab larvae and insect larvae, but they're minuscule. You really can't see them without a microscope. But they would be zooming around, and they'd have legs and gills, and they'd just be moving all over the place. You had to move it as they moved if you wanted to follow them. It was really fun. And they were usually a lot larger. And then the phytoplankton is anything that has to do with plant matter that was growing. So things like kelp that was little, very tiny. And so phytoplankton was really what we were going to be focusing on, but she wanted us to see everything the first day after we collected it. But then the next 2 days, we just looked at phytoplankton. So she gave us a big sheet, and it had a picture of each of the types of phytoplankton and a name under it. And so what we were supposed to do is we took 3 different samples from that same bottle we had collected, and we recorded how many of each type we saw. So it was actually a good thing if we didn't find any, particularly of the bad ones. So the reason they're doing this research is that they're looking for what's called harmful algal blooms. And so I don't know if any of you have seen this before, but there's been places, like I've gone to the beach and it's closed because there's this red algae that's taken over the beach. And well, more like the water, but then washed up on the beach. And you can't go in because it's actually toxic in that large of amounts. So they're concerned because around the world, there has been a very large increase in the amount of harmful algal blooms. So that's algal is algae. And they're trying to figure out why that is, like what's participating with it. So if they can figure out early before it starts, they can take precautions to try and get rid of that algae in whatever means necessary. That could be chemical or natural. She didn't really go into how they would treat that, but what they're trying to do is do research to see what trends they can find, at least even in just California, and then see how that can correlate to other places around the world that are having trouble. So it was a good thing we didn't find many of the bad phytoplankton, but we saw a lot of the good ones too. So it was really cool. We actually took pictures. We would look under the microscope, and then we could use our camera lens to zoom in through the microscope lens and see them. So we have some pictures of that. So part of our goal in being part of the Jason project and going to the Catalina Islands was to help the researchers with their research. So instead of them having to do it every single day, we were participating with them in collecting the data and entering it for them. And it was notated that we were a student group that was doing it.